record. And so then we'll come back and we'll do that other bro science on Wednesday. Okay. All right. Y'all can see here, my Zoom folks, y'all can see?
Yes.
Okay. Y'all can hear me. It's okay?
Yeah.
All right. All right. So we're going to move a little bit away from the idea of talking about health, and we're going to start talking a little bit more so about fitness. But to understand, we're going to start off by talking about muscular fitness, okay? Muscular fitness. So in order to get to the place where we can try to process and understand what happens from a resistance training standpoint and how big your muscles are, how strong your muscles are, what those things let you do, we first need to revisit a lot of what do we need to know about basic anatomy and physiology of skeletal muscle. Okay? So we're going to spend several days talking about kind of how muscles are organized, how they generate force, how your brain communicates with those muscles to tell them to do things, and we'll work through a lot of that stuff. So my hope is that a lot of this is reviewed. A lot is going to be reviewed. But if I'm going to go to the trouble, and this thing is annoying the hell out of me, what I'm hopeful of is that okay, hopefully this will be better, is that you guys will learn the details of all of this. Because what you're going to see, especially kind of at the end of all of this, is that how your muscles are structured. The anatomy plays a role in the actual physiology, okay? Anatomy is structure, physiology is function. So we need to know, so a man here on the end, your name is Ray. We need to know, Ray, how many push-ups can you do? You don't have to get up and do push-ups for us yet. But just like a guess, like how many push-ups? Can you do 10 push-ups? Okay. If Ray's going to do 10 push-ups for us, but he can't do 11, we're going to learn how the structure and function of the muscles that he needs to use, how is the structure and function of the nervous system as his brain communicates with those muscles. How does all of that work to allow him to do that number of push-ups, but not more, okay? Or to do push-ups with a certain type of form that is, I'm hoping for Ray, good, not like me and bad or something like that, okay? So we're going to get through a lot of these. Basic structure and organization. We'll talk about the sliding filament theory of contraction, which I'm sure all of you guys have heard of before. And then we'll get into the nervous system side. So we'll talk about structure, location, function of motor neurons and motor nerves and those things, and how the brain communicates and tells muscles to do something or to not do something. Okay? All right. Okay. So maybe some of you guys have this up and you're going to cheat, but we'll do it this way. Purple sweatshirt. Your name is? Kara. Kara, what is the function of a muscle? It has one job. What's it supposed to do?
Well, if I already looked at it, do you want
That's okay. Just read from your slide.
Generate force and allows for movement.
Okay. Muscles have one job, generate force, full stop, end of discussion. That is it. Okay. That is it. They generate force. No force, no movement. Okay. No force, no movement. It's this generation of force that allows for movement that gets us physical activity, right? It's this generation of force that lets us walk or run or lift weights or whatever it is that allows for exercise. It's the generation of force that burns calories, which gives us what the intensity of that kind of movement is going to be. And then side note of it is you burn the calories and you get to eat more Doritos and more Chick-fil-A or whatever else it is that you particularly like, if that's a thing. Okay? Okay. So we'll move across. White sweatshirt in the back. Katie. Are you generating force right now? You sure? Are you sitting up?
A little.
A little, okay. Are you're wiggling your fingers? Is that force generation? Okay. How are you doing that? Are you thinking about doing that, or are you just doing it? You're just doing it. That's that's a little bit of a trick question. Okay. But you know you need to sit, you have chosen a posture at which to sit. You may not be thinking about it because you may be like me, and I get a little nervous and I start twirling things in my hand and I just kind of do it subconsciously. But in doing that, you're generating force. You are generating the right amount of force to do whatever it is because your brain is signaling to those muscles through motor neurons in your spinal cord to contract. Okay? These muscles contract. These other muscles relax. Okay? The nervous system is the on-off switch for your muscles. You ever had a cramp? Anybody ever had a cramp? Okay? Did you really want it to go away and it wouldn't? Right? That's a muscle contraction that is not controlled by motor neurons. Right? You want to be in a state of relaxation and it's just going anyway. Okay? Otherwise, it's all nervous system, which is good for us because we can teach the nervous system when to turn on, when to turn off, and then to what extent should it turn on. Okay? You will find that this idea of voluntary control, voluntary means you get to choose. Okay? We have a tremendous ability to generate very small, very fine gradations in force production that allows you to write, okay, without smashing your pencil into the paper, okay? All of these things are made, again, to more prescient when you have a toddler. She tries to color. She can hold the thing correctly, but she has a hard time with some fine motor skill things. But you learn over time. Her nervous system hasn't fully developed enough yet to be able to do all of that. Okay, so we'll talk some more about kind of what goes on. But there are very particular structures that let us do this. Okay? Why are muscles and neurons important during exercise? Okay? Let me pick someone. Blue water, black cortisone. You are Hensley. Hensley, can you do a handstand? Okay. Are there things that you can't do? Can you ride a unicycle?
No.
Okay, but you can do a handstand. Okay. So Hensley is there somebody who can't do a handstand? Okay, most of us. All right. So what it requires to do a certain exercise, to do a certain thing, okay, is very precise control, very much on the right amount of force at the right time, okay, in order to let you do something. It's very complicated, right? To do a handstand, Hensley probably has to contract 40 or 50 or 60 different muscles at just the right amount of force, at just the precise right time, okay, to be able to generate that movement pattern to do that one thing. Walking is an amazingly complex motor skill, okay? Lots of muscles at just the right time, at just the right time, okay? If you don't think it's complex, then we'll do a fun thing on Wednesday. And if I have anybody in class that's 21, and we'll give you several drinks and we'll watch your ability to do those motor skills decline, okay? It doesn't take much. Force production is off just a little, okay? Timing is off just a little and you fall down. Okay? So muscles and neurons are important for these reasons. This is very, very complex kinds of things and we don't normally think about that, but it's really complicated, okay? And so by the end of all of this, what I hope is that you'll have an appreciation of how structure and function or anatomy and physiology work together to allow us to do all of these things that we're going to do. Okay? All right. So we're going to start in some ways at the very beginning. We're going to start big here, then we're going to work our way down into kind of small cellular and organelle level structures. So there are three primary classes of muscles in your body, which I'm sure you are aware of. Okay? There's what we think of as skeletal muscle, which is going to be striated or striped. If you look at it under a microscope, it has this very distinct striping pattern. That pattern is due to its structure. It's due to the organization of actin and myosin filaments, and sarcomeres, and things that I'm going to show you guys in there. It looks a certain way. Skeletal muscle is a voluntary muscle. Most of the time, you control when skeletal muscle contracts. Okay? You control. You have somewhere north of about 600 skeletal muscles. Okay? Similar to skeletal muscle is cardiac muscle. Okay? Cardiac muscle is your heart. It looks like skeletal muscle. If you look at it under a microscope, for the most part, it's striated. It looks very much just, it's organized in the same way as skeletal muscle, but cardiac muscle tends to be involuntary. Right? None of you are thinking right now as you're sitting there, make my heart contract, make my heart contract, make my heart contract, right? Stop, slow it down. You're not thinking about it in that way. Okay? It's going to have this involuntary control that comes from the nervous system and endocrine systems, and we'll talk a lot more about that when we get into cardiovascular systems in a moment or so. Okay? But it looks a lot. Then the last kind of muscle is smooth muscle. You have smooth muscle around blood vessels. You have smooth muscle that surrounds your digestive tract. Okay? Smooth muscle under a microscope does not look like skeletal muscle or cardiac muscle. It is a hot mess. Okay? Its fibers run in all different kinds of directions, but because of that, it has a very different job. Okay? When it generates force, we're using it to control the diameter of blood vessels, to make blood go or not go to certain tissues. It's helping to squeeze in a rhythmic fashion and move food or things through your digestive system. Okay? That's what smooth muscle does. It is also involuntary. We'll talk a little bit in the cardiovascular system about how we tell it to contract. We're not going to talk about digestion at all. Okay? Questions about any of that? We okay so far? You see nods of heads and yeses. Okay. Good deal. All right. So, does anyone know what a Russian nesting doll is? Anybody know what a Russian nesting doll is? Your name is?
Casey.
Casey. You were nodding your head yes. What is a Russian nesting doll? Describe it for us.
It's like there's a big one and they're kind of like eggs, and every time you open one, there's another one inside it.
Okay.
Until it gets really small.
Okay. And what do they look like? Like, what is each one as you move down? What do they look like? I mean, they're smaller, but otherwise, is their appearance different than the bigger one? So a Russian nesting doll is like this series of increasingly smaller versions of the exact same thing, right? You got a doll and you open it up and there's a smaller doll. You open that one up and there's a smaller doll. They're all the same, and then they all, like eggs, kind of fit in together, okay? Skeletal muscle, to me, okay, is basically a Russian nesting doll. That's what it is, okay? If you look at it, we go from whole muscle, okay? We cut open your muscle. If you guys had anatomy here, you get to see muscles. You guys get to actually like do the dissection and open them up and cut through the skin and subcutaneous fat and the outside fascia and begin to open them up. So you have a whole muscle, okay? It's got its own connective tissue that sort of puts a sheath around it. You open it up and your whole muscle is made up of these smaller structures that look like whole muscle, but they're just smaller, and they're called fascicles, okay? You open up a fascicle, muscle is a big tube, fascicles are these smaller tubes that are in there. You cut the connective tissue around a fascicle, you open it up, and you got a bunch of muscle fibers. And then you cut open a muscle fiber, and it has all of these little tubes that are myofibrils that are inside of them. And then you cut open a myofibril, and all you see inside of a myofibril are these tubes with these strings of proteins. Okay, so this is the general organization of what a skeletal muscle looks like. You've got the whole muscle, which is the thing that we see most of the time with our naked eye. You open it up, and there are these bundles of muscle fibers that make up these fascicles. We've got a fascicle, and it runs for a ways, and there's another fascicle, and another. So then we come in here, and there's going to be, each one of these represents a fiber. You pull out one fiber, and the fiber has its own little tubes that are myofibrils, and they have these little tubes of proteins in it. Okay? So skeletal muscle is, in essence, a network of these little tubes that are all going to fit together. Okay? What I want you to know about all of this is that a fiber, the muscle fiber, this represents an individual muscle cell. Okay? Muscle fibers are some of the largest cells in the known world. Okay? You can see an individual muscle fiber if somebody would let me sort of cut you open. You can see a muscle fiber with the naked eye. Okay? Some of them may be several centimeters long. They're not very wide, but they're several centimeters long. You can see them with the naked eye. Okay? So these are these giant individual cells that are going to be there. And so what tends to happen is we don't ever really talk about fascicles. We rarely talk about myofibrils. We either talk about what happens with the whole muscle. It's doing a shortening or lengthening contraction. Or we talk about things happening at the fiber level. This is where a lot of cellular physiology comes in on the fiber level. Or we talk about what the proteins in the sarcomeres are doing, because it's the proteins in the sarcomeres that generate force, that actually move. And so the sarcomere moves. And as it moves, the myofibril moves. And as the myofibril moves, the fiber moves, so on and so forth. Okay? So this is this general organization. But it's just all of these tubes within a tube within a tube within a tube. Okay? And things that happen at each one of these are going to be important. Question? No. Okay. I'm going to send you home for arts and crafts time. You got to make me a Russian nesting doll, right, of all of this for next week. You don't have to. That's what I've been home with my daughter. We do a lot of kind of artsy, craftsy kinds of things. It lasts for about 30 seconds, and then she has the attention span, of a puppy. All right. What I've done now, we're going to move away from whole muscle. We're going to start working our way down into smaller and smaller structures as we work down onto the cell level and kind of the subcellular kind of protein level of what all is going to go on. What you see here is a cartoon representation of, we've cut open one individual muscle fiber. Okay, so this is a piece of a muscle cell. Okay. Things that I want you all to know about this fiber because it's important. Okay. I want you to know what the membrane or the cell membrane that goes around this fiber is called. It's called the sarcolemma. All right. Sarco is Greek or Latin or something. But basically, sarco means muscle. Okay. So you'll see a lot of things that are going to say it's the sarcolemma. It's a cell membrane like all other cell membranes. It's a lipid bilayer. It functions just like them. It has proteins in them. It has glycosylation little sites on them. It has receptors that are going to be on it, but it's called the sarcolemma. Okay, so the outside of our fiber, that's the sarcolemma. And it is really, really important. It's really important. If the sarcolemma doesn't function correctly, no muscle contraction. Okay, no muscle contraction. Along the sarcolemma, there are tunnels. Okay, think of the sarcolemma as a road. Okay, as a road. And at various points along that road, there are like manhole covers that you can pull off, and then you can drop down these pipes basically into the sewer. And now we're at Teenage Mutant Ninja Turtle movie or something. Right? It goes along like that. That's what a T-tubule does. Okay, they are these holes in the sarcolemma that lead into a pipe or a tube that runs down into the inside of that muscle fiber. Okay? So those are denoted in our picture here in gold. So here's the opening, and then there's a tube that runs down, and it's going to run down into the inside of our cell. So we use the T-tube to help us take a signal from the outside of a fiber and bring it down into the inside. If your T-tubules don't work right, you cannot get a muscle contraction. Okay? Let me pick my, in the back, in the kind of blue-green, you are Megan. Would you guys like for me to do something to Megan and make her T-tubules not work right? We can do that. No? We could just make Megan stand with her arm out like this, and by the time your arm gets to the place where you're going to have to put it down, we will have made, in your delt muscles and those things, we will have made it where the T-tubules cannot repolarize, and so you can't transmit electrical signals down the T-tubules to do those things. The T-tubule is one of the primary sites where muscle fatigue occurs. Okay? So they're really, really important. Okay? We'll talk much more about muscle fatigue later on in class. We're not going to make Megan do things. Although one time in class, I have had people hold their arms out and said the person that can hold them out the longest would get like 2 bonus points on a test, and it makes for really fun, kind of fun days in class as people sort of struggle to overcome fatigue. Okay? All right. Sarcolemma, T-tubules, we're moving from outside in. These dark kind of maroony structures here that are going to sit adjacent to the T-tubule and then spread out like webs or a net around the individual myofibrils. That's something called the Sarcoplasmic Reticulum. Okay? The Sarcoplasmic Reticulum. You guys may remember from zoology or something, endoplasmic reticulum. This is basically just a muscle's version of some of this. The sarcoplasmic reticulum is just these giant sacs that are full of calcium. Okay? They're full of calcium. And they are connected to the T-tubule, and they are going to exist to hold the calcium, have it inside, and then release that calcium when the T-tubule tells it to do so. We'll talk about probably on Wednesday, kind of all of the mechanics of that. Okay? That's what the sarcoplasmic reticulum is. These three things play a very, very, very large and important role in telling a muscle either to contract or to relax. Okay? That's what's going to go on. Our other two structures, we have these kind of green and yellow structures here that are mitochondria. Okay? I'm sure you all know what mitochondria do. Their primary job is to make energy. Right? They are there to make ATP. They use oxygen, and then they're going to break down fat or carbohydrates or something, and they're going to make ATP. As well as we're going to give off some food. But you have mitochondria in and around all of the myofibrils inside of the fiber. Okay? Mitochondria are really important because we need ATP in order to generate force, or at least to generate force repeatedly. Okay? Every one of you has a different amount of mitochondria. Different size, different numbers, and maybe a little bit of different function in the mitochondria in your individual muscle fibers. Okay? We will talk also on Wednesday about different types of muscle fibers. Not all fibers are made the same. There's some different flavors that they come. Different flavors have different amounts of mitochondria. If you have more mitochondria in your muscles, you are likely going to be a better endurance performer. Okay? You're better at running at a longer distance or cycling at a longer distance or something like that. Okay? We'll talk a huge amount about metabolism and how that works and how that plays into sort of aerobic exercise performance and aerobic fitness and those things. I want you to know that they exist here so we can make the ATP, and then it doesn't have to go very far because the ATP is going to be used by the proteins, okay, that are going to be held in the sarcomeres in these myofibrils. So we want it to be close. Okay? The last thing is what you see on the outside over here in purple. That's the nucleus. Muscle cells are unique because they have more than one nucleus. Okay? Muscle fibers are multinucleated, which most cells in your body, most single-celled organisms like bacteria or protist or things, they only have one nuclei. Okay? They're also unique because their nuclei are on the outside. Rather than being down in the middle of the cell, they're on the outside. Okay? So one of the reasons that muscles have to have many nuclei is they're so freaking big. Okay? The cell itself is so big that one nuclei could not make enough. Okay, it can't make enough protein. It can't sort of, all the nuclei code for the same gene, but they have all the things it tells that muscle fiber, it makes all of the things that it needs for that fiber to function correctly. All of the proteins, all of the receptors, all the other kind of signaling molecules, it makes all of those things. And one nuclei can't get the job done. Okay? There's a reason there aren't giant bacteria in the world. They're not like these huge things, because it's called surface area to volume ratio. And the one nuclei can't make enough stuff to support it being super big. So we get around this in fibers because they're so big by having more nuclei. They're on the outside, which is an interesting thing. We don't really understand why that's the case. But what it allows is we're not taking up space on the inside of the fiber with the nucleus. So we can put more actin and myosin, more sarcomeres, more myofibrils on the inside. So it gives us an opportunity to have as much protein that generates force as possible taking up the inside of that fiber. Okay. Does that make sense, guys? You all understand? Okay. Those are a few of the things. We don't in this class talk too much about the idea of transcription and translation and the role that the nuclei play in synthesizing new proteins. We'll talk very, very briefly about a few genes that may play some role in making muscles get bigger when you resistance train. But that's really going to be it. Dr. Mike in Exercise Phys will cover that in a lot more depth. Okay. Okay. So what I want to point out are, here's an electron micrograph. And I apologize in here with our lights, it's kind of hard to see it that well. But this is one muscle fiber. And you can see this pattern, the striping that's going to take place in there. And what they have done is they've stained this. They've stained it so that the fiber itself looks red. These kind of green yellow spots on the outside are nuclei. So this fiber has many, many nuclei. But then it also has on the outside these blue structures that look a lot like a nucleus. But they are, in fact, what we call satellite cells. Satellite cells are muscle-specific stem cells. You guys know what stem cells do? Anyone heard of stem cells? Anyone? Anybody know? Okay. You all are aware that there are these things called stem cells. Yes? Okay. Stem cells are important because depending upon where the stem cell is from, or if you can get them at certain stages, stem cells have the capacity, because they are undifferentiated, to potentially become any kind of tissue that we want it to become, if you can give it the right signals. Okay? So if I have a stem cell and I give it the right signals, I could grow you muscle, or I could grow you a liver, or I could grow a kidney, or I could grow, a peripheral axon or something out of these stem cells. Okay? So these satellite cells are muscle specific. They're only ever going to become muscle fibers. But muscle is also unique in that they have their own cells that are kind of right there with them. Not all tissues do all of this. And we know that satellite cells play a really, really important role in repair and recovery of muscle after exercise, especially after resistance training. Okay? I could take one of you and I could give you a certain kind of radiation, and I can kill off all the stem cells, all the satellite cells. I kill off a lot of them. I can kill off the satellite cells in the muscle. If I then took you and had you do resistance training, your muscles will not get bigger, hardly at all, from doing this. Okay? We've never done this in a human as far as I am aware. They do it in animals all the time. Okay, you irradiate the satellite cells, you have them do exercise, even if you give them, testosterone and growth hormone and all these things, their muscles still don't get big. So we know that they're really, really important. We activate them because you damage your muscle fibers all the time. Okay. Anybody sore? Anybody sore today? Okay. If you're sore, you have damaged skeletal muscle that has evoked an immune response. If you are sore, your force production is probably a little bit lower than it was yesterday, the day before, whenever you did whatever you were doing to make yourself sore. I am sore because I built my daughter a rock climbing wall this weekend as part of her birthday. And we had to carry it across. We were hiding it at our neighbor's house. We had to carry it across the street. When you're old like me, doing things like that make you sore because you're out of shape. But I have damaged muscle fibers. They are being regenerated right now as we speak. Okay. Part of that regeneration process is we tell the satellite cells to turn on. We kill off the damaged fibers. We make new ones for the satellite cells. Okay. This happens all the time. And as the satellite cells proliferate, they split, and one of them stops and kind of becomes a satellite cell and stays one, and the other one becomes a new muscle fiber. So some really, really cool things. The Dr. Mike, again, will go into more depth about all of those in an exercise space. Okay. All right. Let's zoom in a little bit farther. Okay, a little bit farther. We've got our muscle fiber. These tubes inside of it are what we call myofibrils. We don't talk about myofibrils very much because they're kind of boring. But what we do talk about is the fact that a myofibril is made up of these structures that are called sarcomeres. You have one sarcomere, another sarcomere, and another sarcomere, and another lined up end to end, and those all together make a myofibril. Okay? So let's talk about what's going on in the sarcomere. The sarcomere is the basic functional unit of skeletal muscle. Okay? Anytime you move, what is happening is that the sarcomeres in the muscle that is shortening or lengthening, it's the sarcomere that initiates that change in the length. Okay? The sarcomere shortens. One sarcomere pulls on another, and we get out to where the whole muscle changes. It pulls on the tendon, and then that pulls on the bone. Okay? So in a lot of ways, the sarcomere is where the action is. A sarcomere is going to be bounded on either side by what we call a Z-band or a Z-disc, okay? Which is this area here and here, okay? Sarcomeres have this very kind of highly specialized structure where you have this small little like three-dimensional cylinder. Along the outside of the cylinder, along the Z-disc, okay, and then along a few things on the outside, you have this scaffolding. These are structural proteins. They're made up of things called desmin and nebulin and vinculin and titin. They're these big structural proteins, but it's like framing in a house, basically, or scaffolding on a skyscraper. Okay, so they're there and they give it this sort of shape and structure. And then on the inside, you have these thick and thin filaments, which are just these long strands or strings of these little proteins that are all hooked together. Okay, a thin filament is made up of the protein actin. I'll show you guys a picture of this in a second. Okay, actin is a sphere. So they're these little balls, and you have one hooks to the next one to the next one. So it looks like a string of beads. And then actin, a thin filament has two of those strands of beads that are wound around each other. Okay, that's a thin filament. A thick filament is made up of the protein myosin. Okay, your muscles are about 2/3 myosin and about 1/3 actin. Myosin, in my mind, is, you can make an argument that myosin is the single most important protein in the body as it relates to physical activity. Okay. Myosin is what we call a molecular motor. Molecular motor. So myosin is the thing that moves. Myosin is the thing that generates force. So the more myosin you have, theoretically, the more force you can generate. The person that has a bigger muscle, or just think about it in yourself, your quads are probably bigger than your biceps, right? So I look around and make sure we don't have, you may have seen some bros who that is not necessarily always the case in. They've never done a squat and do a lot of curls, right? Big arms, little tiny legs. Okay, but in general, the bigger the muscle, the more myosin it has. The more myosin it has, the more individual proteins that can generate force there are. And think of it, more myosin, each one generates X amount of force. Just add up all those myosin. If you've got more, then we can roughly expect they can generate more force. All right, so I'll show you guys in the next couple of slides a little bit more of how the actin and myosin are going to look by themselves, how the thick and thin filaments are going to fit together, and how three-dimensionally, right, this sarcomere is organized to allow us to stack in as much actin and myosin as possible in this kind of three-dimensional space, okay? Which means we have the capacity to generate a huge amount of force, okay? So here's a little bit higher resolution look at my individual cells, okay? So we have Z disc here on the right, Z disc here on the left, okay? Anchored to the Z disc are going to be our actin or our thin filaments. Remember, I told you guys, actin is a sphere, and then these strings of beads, and they're wound on each other. So you've got these actin filaments. They're going to extend from the Z disc out towards the middle, okay? And then interspersed kind of alternately between all of our actin filaments are going to be our myosin filaments, or our thick filaments. So myosin, in a lot of ways, it looks like this, okay? It has what we call a head region and a tail region, okay? People have said before, it sort of looks like two molecules of sperm where their tails have wound together. That's fine, okay? It is whatever it is. You've got the binding site here on the myosin head, okay? And the heads are going to protrude off at like 60-degree angles. The head region binds onto a location on the actin filament, okay? It binds and then it pulls, okay? So this end will tilt and it always tilts in towards the middle from both directions. So it binds on, it pulls. As it pulls the actin filaments, that pulls at the Z disc. This sarcomere then squishes in towards the middle. It pulls the adjacent sarcomere with it, which pulls the adjacent sarcomere, so on and so forth. And that's how we do that. Okay? So in the thick filaments, the tails are all going to be kind of wound up together to generate this kind of loose filament structure, and then the heads are going to protrude off. Okay? Three-dimensionally, okay, this is, you're just kind of seeing two dimensions. If you think three-dimensionally, if I take this and I turn it, and you're looking down into this right end, what you see is it forms a hexagon, okay? There will be a myosin filament in the middle, and then at each of our sort of 6 points around the hexagon, there will be an actin filament, okay? And then on those, there will then be another myosin filament. Okay, do I have a picture of that? No, it appears that I don't. I'll back up. You can sort of see it here. You've got a thick filament, and then 1, 2, 3, 4, 5, 6. You've got these 6 actin filaments around it, and then another thick filament here, and it's going to have 6. And so we're sharing some of these among several thick filaments. This arrangement like this lets us pack as much actin and myosin in a small space as we possibly can. Okay. Couple of last little things here from a structure standpoint that I want you to know. Right? We already talked about earlier, right, that Katie did not have all of the muscles in her body doing a muscle contraction right now. Is that fair, Katie? We still that way? No cramps? You're still doing all right? Okay. So, the normal state, okay, of actin and myosin is that they are not connected and they are not generating force. Okay? That's the normal state. Well, the problem is that myosin really likes actin. It really likes it. It really wants to bind to it. It's really kind of creepy. It really wants to pester you. Okay? So we have to have some things in place to prevent the myosin from binding to the actin. So there are two what we often refer to as regulatory proteins that exist on the actin filament that I want you guys to know. Those are called troponin and tropomyosin. Okay? So if you look at the picture, tropomyosin is in pink on our picture, okay? And the troponin is in kind of yellow or gold. And so tropomyosin is a long, thin protein. And tropomyosin, it's like those little plastic covers you put into light sockets to keep kids or your drunk friends from like sticking their fingers in. Okay? That's what tropomyosin does. Tropomyosin sits over the top of the myosin binding site on actin, and it prevents myosin from being able to bind. Okay? It sits there and keeps the myosin from binding. Troponin is going to be connected at various these kind of regular intervals onto the tropomyosin. And troponin does sort of two things. Okay? Troponin binds calcium. Calcium that has come most likely from the sarcoplasmic reticulum. But it binds calcium, and when it does, anybody had biochemistry? No one has had biochemistry. They may know where you have to, we make all of our PhD students take biochem. You now have to have organic if you want to take biochem. If you're in here, which as a person that has had both organic and biochemistry, terrible. I've never used organic in my entire life, ever. I never had a class in anatomy. Did I tell you guys on the first day? I've never taken anatomy. I've taught anatomy and physiology, and I've never had anatomy. So we'll see how that goes. So if I tell you things, maybe don't believe it. So troponin undergoes something called a conformational change. And what that means is calcium binds to troponin, and the troponin changes its shape. It's like a transformer. Okay? And as it changes its shape, it sort of moves in a way that pulls and lifts the tropomyosin off of the myosin binding site. What happens is if I want a muscle contraction to happen, I bind calcium to troponin. It undergoes its conformational change. That pulls the tropomyosin away. And then the myosin now is like, what's up, actin? How's it going? Right? It binds, and then once it binds, we get what's called a cross bridge and what's called a power stroke, and that's how we get force. Okay? Then the myosin breaks off, and calcium goes off of troponin. It goes back to its original state. It puts the tropomyosin back on the binding site, and now you've relaxed that individual contraction. That's kind of what I was saying. Okay. We'll talk more about cross bridges. We'll talk more about what happens on the myosin head. There's a thing on the myosin head called the myosin ATPase. It's an enzyme. This is the thing that uses energy. Okay? And how all of that works. Okay. All right. Let's pause. It's 11:30. Let's pause for a few minutes, and then we'll talk about muscle fiber types, and we'll get you guys out of here. Okay. All right, we'll resume recording. Okay. How many of you guys have heard of muscle fiber types? Okay. On the very end, gray sweatshirt, your name is? Kenzie. What have you heard about fiber types? Okay. Correct. All of those things are. Okay. So that's pretty good. So if we're going to talk about, as I said, that muscle fibers come in these different flavors, these different types. in order to understand how we learn that this was the case, we have to learn a little bit about something called a muscle biopsy. Okay? So when you biopsy a tissue, you take a piece of it out, and then we look at it under a microscope or something, you do something to it, and you can learn things about that individual tissue. So the specific technique that is most commonly used to take what's called a muscle biopsy it's called the Bergstrom technique. And it was, unsurprisingly, if you know anything about names, it was developed in Scandinavia. It was developed by a Swedish fellow whose last name was Bergstrom. And so when we do this, and so the lab that I worked in as a PhD student in Georgia, we took muscle biopsies. To take a biopsy of your muscle, you make a small, about a 1 centimeter incision in the muscle, and you cut through the skin, the subcutaneous fat, and then the sort of the fascia that surrounds. We're going to stick the needle down into the fascicle. Okay? The needle is a pretty big needle in comparison to like what you get in an actual shop. Okay? It's not quite as big around as a pencil, but it's not that much smaller than that. You stick the needle in, and it looks funky. Okay, it has the end of a needle, but there's not like a hole on the end to inject stuff. It has this little window, and the way that it works is you have an outer needle, and then you have an inner needle, and you connect these two things. It works on like a little plunger. Okay, so the needle works like a guillotine. So it opens this little window on there, and you suck a piece of muscle into that window, and then you slide the inner needle down, and this kind of leading edge of the inner needle here is essentially a razor, and it cuts off this little piece and it gets stuck. So you put the needle in, you hook a plunger onto this end, and you put suction on, and that pulls a piece or pulls some individual muscle into the window. Plunge this end down, it cuts a little piece off, you pull it out, it looks like this. You mount it on a little tongue depressor, you dip it in some liquid nitrogen and freeze it, but it ends up looking, it's about the size of the eraser. It's an eraser on the pencil. It looks this way. And then you take all of that, and some poor grad student like me has to put it in what is the best cutting, basically deli slicer you've ever seen in your life. And you take these little tiny thin individual slices on the end of it, and you mount those. They're translucent, they're so thin, and you put them on a slide, and then you can bathe the muscle in different things to show the fiber type, to show how many mitochondria it has, to show how much glycogen is in there. And you can then look at those under an individual microscope. Okay? And then you can do other things. You can take a biopsy, and we can look for what's called messenger RNA. So if we want to know, you did some resistance training, we want to know how much myosin messenger RNA are there. So how much has this thing been told to make some new protein? All that kind of stuff afterwards.
Didn't you get in trouble for this back in the day? Didn't they were doing something wrong?
How would you know that?
And I took principles of endurance training, like John actually was talking about it.
Yes, we did get in trouble. And I say we. Well, I am here because some people got in trouble doing this and were not so politely asked to leave and go find other jobs. Yes. But it was not that they were taking the biopsies incorrectly or that they hurt anyone. It's that so when you do research, you have to have something called, what's called an institutional review board. And you submit your research protocol, and they say, we think this is safe. We think that you're doing this ethically. This is going to be okay. And you get permission to do it. And they were taking biopsies when they weren't supposed to be. And there were some people, I think, that felt like maybe graduate students of those individual professors that felt like they didn't have the option to turn down having to give up a biopsy in that way. Because I can tell you, the actual biopsy doesn't hurt that much. You just inject the site with a little bit of local anesthetic. So it's just, you get a little bit of local, you feel some pressure, the needle goes in, and then you close it up, usually we would just put little butterfly clips on there, But what happens is like the next day and the day after, like it hurts, it's sore from all of that. And so, you know, it's, I mean, it is considered like, this is like surgery in some ways, if we're getting down into tissues. So there's some potential, if the person doesn't know what they're doing, they're going the wrong place, you can hit a nerve, you can hit a blood vessel, like there's some really kind of downside things. So I gotta go talk to John about what he's telling people.
I just couldn't remember.
No, but
it was
that's
nothing bad.
no, we did. And so because of all of that, like we've not taken biopsies here since I have gotten there. And I think we could, but we would have to probably get an MD to come down from the health science center and actually do the biopsy for us. They probably wouldn't let us do it. So you are, run me of your name. Abby, what you got?
So what you find in that biopsy, is it going to be consistent in all the muscles throughout the body?
Great question. Great question. Okay. So you take this biopsy. This one, they're taking it from a person's calf here. You can take them from the calf, and you can theoretically take them from any superficial muscle that you can have access to. Most commonly, it's the calf or the vastus lateralis or the bicep. Okay. What we find in that muscle is specific only to that muscle. In some ways, it's only specific to the little area in the muscle where the biopsy came from. However, what Abby's muscle from her vastus lateralis looks like is going to be closer to what her calf and her bicep looks like than what something potentially from Ray's leg or arm is going to look like. Okay? There are differences among muscles. They're subtle, but they are different. Okay? All of you have a genetically determined percentage of your individual muscle fibers that are type 1 and that are type 2, either type 2A or type 2X, which we'll talk about in just a second. That is genetically determined. Okay? What we tend to find is that muscles in your body that you use most often, okay? Gastroc, soleus, okay? Diaphragm, your erector spinae, okay? Postural muscles tend to have a higher percentage of type 1 fibers than non-postural muscles in your body. So your calf is probably going to have more type 1 or slow twitch fibers than the quad is going to. Your quad may have more than your bicep does, okay? But we'll see a little bit of that as we go through that. Okay. Okay. So there was a time, let me back up. This came out in the 60s. Okay, this came out in the 60s. In the late 60s and early 70s, there was a revolution in biochemistry. Okay, where a lot of the things that we do now, like when you go and you take a rapid test for COVID, and it does the chromatography as it pulls things along, they have embedded what are called monoclonal antibodies in there that will turn a certain color if it interacts with an antigen from COVID. Or if you go and you get the PCR test done, all of those techniques began to be developed in the late 60s and early 70s. And so what we figured out was we could begin to look at the individual kind of characteristics of enzymes in each of these muscle fibers, and that they were a little bit different. In the amount of glycogen that was in each muscle fiber, in the amount of mitochondria that were in each of these individual muscle fibers, because of the advent of monoclonal antibodies and what we now think of as kind of modern molecular biochemistry. Okay, so these two things work in concert, and we know a lot of things about kind of what happens with muscles, with training, what athletes versus non-athletes look like because of all of that. I mean, a lot of that stuff has really gone out of favor. We don't, there's nothing else for us to learn as we do all of it. So initially they would pull out these fibers, and we classified that we noticed that these individual fibers differed on the speed at which they would generate force. Okay, this was called contraction speed or contraction velocity. And so we separated fiber types based on those that generated force quickly and those that generated force more slowly. And I apologize for my folks on Zoom, but if you have an individual fiber, okay, and so what we have here on the x-axis is time, and on the y-axis is force. When I make that individual fiber contract, as the actin and myosin bind together, you're going to get a cross bridge, you get force. There's a little bit of a time lag on all of those things. And there's a little bit of time lag in how quickly, think of it as like pulling slack out of a rope. Okay? It has to pull all the slack out of the connective tissue and things in your muscle. All right. And then once you pull the slack out, then you can actually try to win the tug of war on those things. And so it looks something kind of like this. Okay. So you get some peak level of force production here, and there is what we would call, it's oftentimes called rise time. From this 0 point up to kind of the peak, there is some amount of time that it takes to get there. Okay. Or you can measure the slope from like here to here, you can measure the steepness of the slope in our rise. And what they found was that some fibers look like this, and then other ones look something like that. Okay? So a very steep, fast increase in force, and then one that was a little bit slow, a little bit more gradual. And so they said, these that go up quickly, we're going to call those fast twitch fibers. And then the ones that are a little bit slower, we're calling them slow twitch fibers. Okay? So before we could do all of the molecular biochemistry, they were looking at the fibers that way. Some of them generate force quickly, some generate them slowly, and we could kind of count up how many of those in each little piece of a biopsy that you have. Okay? Once we got better at doing biochemistry, they were able to look at something called myosin heavy chain isoform, or MHC. Okay? MHC, myosin heavy chain. I'm not going to get into what the myosin heavy chain is and what the myosin light chain is and how all of those goes, but this is, in essence, it's a protein that exists on the head of the myosin protein. It's a small little protein on the head of the bigger myosin protein. Okay? And what they found was that there were a variety of these MHC proteins. Generally, the three most common ones are what we call type 1, type 2A, and type 2X. Okay? Type 1 MHC always occurs on fibers that have a slow contraction speed. Okay? And then on the fibers that had a fast contraction speed, we tend to see either type 2A or type 2X. Okay? So people oftentimes will use these terms interchangeably. They'll say, I got a fast twitch fiber, or I've got a slow twitch fiber. Okay? And that's fine. But if you say a slow twitch fiber is also called a type 1 fiber because it has type 1 MHC, fast twitch fibers, within that fast twitch, there can be several individual isoforms or several individual fibers. Most commonly going to be 2As or 2Xs. It's a little more complicated than that. Dr. Mike will talk about there are fibers that exist in between those, these kind of hybrid isoforms and things, but it's a little beyond what I want us to know. Okay? 2A versus 2X. 2As are a little bit slower than 2Xs. They generate a little bit less force, and they're way more energy efficient. They have an ability to make a lot more ATP because they have more mitochondria. Okay? Think of a type 1 fiber as a Prius, or it's a Tesla, right? 2A is a nice kind of like sedan. It gets pretty reasonable gas mileage. 2X is a Hummer, right? You get like 3 miles to the gallon. Lots of force, but terrible gas mileage. Okay? So in your muscles, most of you are going to be somewhere, one way or the other, about 60% to 40% type 1 versus sort of the collection of 2, so fast to slow. You're maybe 40% slow, 60% fast. Or you're 60% slow, 40% fast. Most of you are somewhere around that area. Elite distance runners are like 90-10, slow to fast. Okay? Elite sprinting athletes, okay, are very likely like 90-10, type 2 to type 1, or fast to slow. Okay? Your diaphragm is the slowest muscle in your body, in everybody. The diaphragm is really, really slow. Okay? It's very fatigue resistant. It doesn't look like the rest of your things. Okay? In a person that is normally like 50-50, your diaphragm might be 80-20, slow to fast. Your soleus tends to be the next slowest muscle. If you're normally 50-50, it might be 60-40. Okay? But in general, most of us are kind of somewhere in that relatively kind of middle, narrow range. This is all genetically determined. You cannot change this. I don't care how much you run or how much you lift, you cannot change the percentage of your fibers that are slow twitch versus fast twitch. Okay? Doesn't matter. What you can change is the proportion of your fast twitch fibers that are A's and X's. Okay? You cannot turn slow twitch fibers into fast unless I give you a spinal cord injury and you're sedentary for like 10 years. You cannot turn fast into slow no matter how much you like. But I can, if you do any kind of activity whatsoever, you will turn your 2 X's into 2 A's. Running, cycling, weightlifting, walking, doesn't matter. Okay? Most of you guys, because you are active, have very, very few 2X fibers in your muscles at all. They've all been converted into 2X. And that's a good thing. That's a good thing. That means they're more resistant to fatigue. If I put one of you guys on bed rest, okay, on bed rest for say 6 weeks, you just got to lay in bed for 6 weeks. I will turn your 2A fibers, some of them, into 2X's because you're very, very sedentary. Okay. There are sprinters that train and train and train and then stop for like 3 weeks and lay in bed sometimes. At least they used to try to turn the A's back into X's before competition. It's a pretty dumb thing to do now because you lose all of the nervous system kind of training that you have while you do all of that too. That's kind of what goes on. Question?
Can you repeat how you said you can turn A's into X's and X's into A's, but you can't turn them into?
Correct. Yeah, you got it absolutely right. Okay. Genetically, you have a certain percentage of slow twitch and fast twitch fibers, okay? Or type 1 fibers versus all of your type 2. Okay? We cannot change that. We cannot change it. What we can change is of your type 2 fibers, of your fast twitch fibers, we can alter the proportion of those that are 2A versus 2X. Okay? So, where's my chart? Let's just say that you are 50% slow, 50% fast. Okay? I can't change this. But when you were born, you were 50% slow, 50% fast. Of this 50%, okay? Of this 50%, let's just say it's exactly also 50% 2A. So in your body, you're 50% slow twitch fiber, 25% 2A, 25% 2X. You guys following me on my map? Okay. When you become active, any kind of activity whatsoever, and your 2X fibers will turn into 2A's. So you're still 50-50, but in that fast twitch 50%, it's now, let's say, 40% 2A, 10% 2x or something like that. Okay? Is that a better illustration? Sometimes I know it's probably helpful to see kind of how this goes. And it doesn't matter. You can run, you can walk, you can bike, you can swim, you can lift weights, anything. The body does not like to have two X's around because they're so energy inefficient. Okay? They're so energy inefficient. Question.
Does that account for the marathon runners or the 90-10 for the slow twitch, or is that a genetic?
Okay, great question. So I have marathon runners that are maybe 90-10, okay? In their 10%, it's probably like 9% A and like 1% X out of all of that. But this person is genetically gifted to be an endurance runner. This person can train like hell and become a lot better as an endurance athlete compared to where they started because they turn their X's into A's. As you guys will see, the 2A fibers can have a huge amount of mitochondria in them. You can make your type 2 fibers much more aerobically sort of inclined. And so I can take every one of you and make you a much better endurance athlete if you'll just do it. If you'll just run or bike or walk. You may never be like a 2-hour and 10-minute marathon. You may never be elite. Whereas there's lots of people that are 90-10 that aren't even elite. But you've got to have this, and then you got to train, you got a bunch of things. But I can make all of you better because of this. But I may not be able to get you way over that kind of other. I can't make any of you better sprinters. I mean, I can make you a little bit of a better sprinter by teaching you form. But if you're not fast now to begin with, I can't make you like super duper fast. It's just not going to happen. Right? You can't jump high now because you have a lot of type 2 fibers. I can't help you. I'm sorry. I can make you a little bit better, but I can't make you way better in the way that I can if you'll just come running. That help?
Dr. Black, I have a question.
Yes, Madison.
So if you're born with like say 60% slow fibers,
Yes.
like can you increase or decrease, like change that at all?
No.
Okay. Just making sure.
No. Your slow fibers are whatever they are. Okay.
Okay.
Now, when we get into adaptations to aerobic training, within your slow twitch fibers and your 2A fibers, you can make the number of mitochondria that you have in them go up, and that helps make you a better endurance person. But
okay,
you can't turn ones into twos or twos into ones. You can't do those things.
Okie dokie.
Other things that we can do in all of this, we can look at the metabolic activity, right? You can look at kind of how many mitochondria. Slow twitch fibers have a lot, fast twitch not as much. On the highly glycolytic, which is more of the anaerobic metabolism, type 2s tend to be more glycolytic than 1s. We can do that. You can stain and look at how much glycogen is in there, which is stored carbohydrate. Type 1 fibers tend to have a lot of glycogen. 2s tend to have a little bit less. So we can do a lot of different things. Okay. The other piece that I want to talk about in this sort of table that just kind of lays out what each one are going to look like. What I want you guys to look at are capillaries, okay, and then power production. Another piece of metabolism is the ability to bring oxygen into those fibers. And so type 1 fibers tend to have more capillaries that surround them than type 2, either A or X. And 2As tend to have more than 2X. If you get more capillaries, then it's easier to get oxygen and glucose and things into those fibers, and that's a good thing. We'll talk about changes in capillaries. Okay. The last little thing is power. If you look back over on the left, okay, power is force multiplied by velocity. If you want to sprint, you want to run fast, jump high, okay, you need to generate a lot of power. And so the reason that people that have a lot of type 2 fibers have more power is that the larger percentage of their fibers generate force very quickly, and those fibers tend to generate a little bit more force on the top end as well. Okay? So people that are good sprinters also tend to be people that can jump higher because it empowers this application of force very, very quickly to let you jump and run and do all this. Okay? And so a lot of that is just genetics. You're either fast or you're not. Okay? Or you're a good jumper or you're not. Okay, so we talked about that. All right, this is a good place for us to stop. Okay. On Wednesday, we'll pick up talking about the nervous system side of things, and then we'll move into actually putting what happens in the nervous system together with what happens in the muscle to get actual force and movement. Okay. All right, if you didn't give me your stuff, I need it. I will see you guys on Wednesday. Thank you.